we're going to do here is just kind of get settled into the environment of metabolism. What are the major biomolecules? What is the environment of the cell? We will also look at our first major disease. But overall, this lecture shouldn't be too complicated. We're just getting started. We're just going to figure out what are the major players in metabolism and what roles they fulfill. So, to begin with, biochemistry and metabolism, they really are two disciplines that are examining how life works in the only way that we can really do that. We're going to look at this in chemical terms, chemical reactions, an entire series of chemical reactions that cover all the major components of cellular behavior, and ultimately those will cascade up to controlling all the major chemical events in an organism. It is important to note that biochemistry and metabolism are not chemistry or a subset of chemistry. Neither are these things biology or a subset of biology. They really are their own disciplines, and they're very, very unique. They draw on a lot of other disciplines, including physics and math and biology and chemistry, but they really are doing something different and looking at a system under very particular circumstances. These systems are examining the logic of life, which, as you can imagine, is an incredibly complex and difficult question. We're going to take something as simple as, say, any protist or even a bacteria and look at it chemically. Imagine we do, say, a double displacement reaction in chemistry in which one material is exchanged for another and we have A plus B yields C plus D. And those can be difficult to examine as well. Now we're going to expand our view to thousands of interdependent and interconnected chemical reactions, all of which are behaving in a vaguely predictable manner, but are really behaving in simply a trend to relieve stress on a system. And we'll get over that in a lot more detail later on in the course. So what I'm about to show you is pretty much the cornerstone molecule of life. I want you to ask yourself if you can identify it. So what is this? What is this molecule we're examining? I'll give you a second to think about it. This molecule is glucose, and it really is the cornerstone molecule of biochemistry. The miracle is that in the first part of the 20th century, there was detailed analysis of how glucose was metabolized in yeast and animal cells. And these experiments found that all the reactions showed the same 10 chemical intermediates and the same 10 enzymes. This is remarkable. And remember, prior to this discovery, it was completely unknown how living things dealt with chemicals, how living things dealt with food molecules, and everything had its own little niche, and we had vitalism at the time, and so there's this special magic that living things had in which only living things could create biomolecules, which turns out to be not true. But then we go a step further and discover that everything processes metabolism in basically the same way. All of life uses the same metabolic systems. Now, there are some exceptions. For example, not everything photosynthesizes. We have the archaeans that do their own kind of weird little thing. But the core of metabolism is identical across organisms. This creates a profound unity to life and further underscores how life descends from a common ancestor. Metabolic systems are astoundingly highly conserved. So, on this idea of conservation, we also can narrow our focus to specific elements. So, we have roughly 90 naturally occurring elements, and life is going to use less than 30 of them. Our big players: hydrogen, carbon, nitrogen, oxygen. Those are huge. We could put in phosphorus as well. and then sodium, potassium, calcium, sulfur, and chlorine. These are your primary elements of life. Now, chlorine's really going to be chloride, but you get the idea that these are the atoms that constitute life. We have some additional atoms that we need, but very, very, very small amounts, what are called trace amounts. These are really small amounts. Those are magnesium, vanadium, chromium, manganese, iron, cobalt, nickel, copper, zinc, selenium, molybdenum, and iodine in only trace amounts. To give you an example of what that means, when we discuss oxygen transfer in heme, that is entirely dependent on simply 4 iron atoms in hemoglobin, and that's it. 4 iron atoms. Now, that's in every hemoglobin molecule, but compare that to the ridiculous abundance of carbon, nitrogen, and especially hydrogen. And you can see that these metals, while you have to have them, there is an absolute bio requirement for cobalt, for copper, zinc, etc. We don't need much of it. To give you an example, let's look at copper. Copper is an astoundingly regulated metal, most of them are, and you consume roughly 5 milligrams of copper per day. And that's what you take in. As it happens, you also excrete 5 milligrams of copper a day. So the amount you intake will equal the amount you excrete. So copper levels are kept constant. There are some diseases we will see later on, Wilson's and Menkes diseases, that are complete disruptions of copper homeostasis. But we'll see those later on in the course. So here's our standard periodic table. And it turns out that the vast majority of it serves no purpose in life. That, as we can see here, only a handful of elements are involved in the processes of life. And similarly, as we will discuss later on in the course, even though there's something around 80,000 proteins, chemical reactions in life really boil down to 6. So we can see life is astoundingly conserved, so all living things do all the same processes, though there are a few bolted on additional processes, and those processes, the trillions and trillions and trillions of those reactions, are all accomplished by basically 6 chemical reactions. And within all of that, there's only a handful of elements that are even relevant to the discussion. So while I don't require students to really memorize structures in metabolism, it can be very, very helpful because the names of enzymes and substrates often do reference structures, and so if you have a chemical structure in mind, the enzymatic reaction makes more sense. But there are some things you do need to know, and those are sort of summarized here as common moieties in biochemistry. These are our big ones: methyl, ethyl, ether, aldehyde, carboxyl, phosphates, esters, hydroxyls, phenols, ketones, aminos, and sulfhydryls, and thioesters. The reason I'm calling these to your attention is that they appear all the time. We will see the amino on every peptide. Carboxyls are everywhere. Phosphates, well, there's ATP. Sulfhydryl, that's cysteine, you'll see that. And so what I want you to be able to do is to recognize these names for the structures that they are, just so that that will help make more sense when you encounter enzymatic names and reaction names. And now to kind of get into sort of the central point of the lecture, and that is our 4 classes of molecules. What's interesting, again, in our limited scope of what metabolism even examines, there are only 4 major classes of molecules. That's it. That's all we're going to look at. We're going to look at proteins. We're going to look at a lot of proteins, nucleic acids, lipids, and carbohydrates. And these are proteins, which are made up of amino acids, nucleic acids, which are made up of nucleotides, lipids, which comprise fatty acids and cholesterol, by the way, and carbohydrates, which are basically just carbon and hydrogen, or, as you're probably more familiar with, sugars. And that's it. And each of these things plays a specific set of roles. For example, there are three main roles: action, where we're actually doing a chemical reaction of some sort; information storage and retrieval, where we're storing information in a chemical form and we need to retrieve that information for use; and of course, providing energy. Under action, we'll see proteins and enzymes. Under information storage and retrieval, you'll see primarily nucleic acids. And under energy comes everything. Every single biomolecule is capable of being used for energy by the cell. Not shown here, but also important to note, is structure, so just scaffolding. They're not action, just structural molecules: cell walls, carbohydrates, plasma membranes, lipids. You get the idea. Cytoskeleton, proteins. Nucleic acids are not used frequently in structures. So let's look at proteins. Well, proteins, they're comprised of amino acids. We get that. There's things like glutamine and glutamate and glycine, etc. They have precise 3D structures, and they have a huge array of functions. Now, this precise 3D structure thing is a little bit of a misnomer. What we see all the time in pictures are these types of structures over here. These are called ribbon structures, and it says, okay, well, the protein looks like this, which is nonsense. The proteins do not look like this. They do not look like their image because they're constantly in motion. This specific image, probably derived from a crystal structure, is just an average of the structures the protein can adopt that will form a crystal. That is completely not biologically necessarily relevant. We need to be very careful about statements like that, but as you can see, they don't have a precise 3D structure so much as they have a series of 3D structures that are all related to each other. This is our standard amino acid structure. You are responsible for knowing this. This is the correct charge. So we have the amine that is protonated, and we have the carboxyl group that is deprotonated. This would be probably a pH 7.4, which is the standard pH that we are going to be using in biochemistry. There are a few regions of the cells we will see that will have different pHs, but in general we can kind of keep a pH of 7.4 in the back of our head. And at that pH, the amine group will have an extra proton, and the carboxylate will have lost its proton. If we drive the pH up high enough, say somewhere around 12, then the amine group will also lose its proton. And if we drive the pH low enough, say 2, then the carboxylate will have gained a proton and would be called a carboxylic acid. Now let's move on to the nucleotides. Nucleotides carry chemical energy. This is ATP. So by far we see ATP as our primary energy carrier in the cell. Now don't confuse an energetic molecule with an electron-rich molecule. So an energetic molecule is going to be something that will produce products that are more stable, and we'll look at that in a moment. An electron-rich molecule is something that is extremely stable, will not produce more stable molecules, but has a lot of hydrogen. As you can imagine, methyl, CH4, and carbon dioxide, CO2, are equivalently stable. They're not terribly energetic molecules. They're very, very, very stable molecules. But methyl has all those hydrogens, which carry electrons. So methyl can be used in a reaction to capture those electrons in a redox reaction, but carbon dioxide could not. So that brings us back to energy. So ATP is our primary energy carrier. It is also an energetic molecule, whereas glucose, by comparison, has a lot of electrons, so it's good for harvesting electrons, along with the fatty acids, very good for harvesting electrons, but are not energetic. Nucleotides such as ATP will also function as signaling molecules. We see these, they can be cyclized to cyclic AMP, or perhaps the ATP itself is just informing the cell of something, that something is happening in the environment. ATP and other nucleotides also function as what are called coenzymes. Coenzymes are not Proteins, necessarily, they're usually not. They're usually metals or small molecules, but they allow a protein to function in a specific way. For example, kinases move phosphates from ATP and put them onto some other substance, but they cannot function in the absence of ATP. They also can't function very well in the absence of magnesium. So magnesium as well would be a coenzyme, but it's not a nucleotide. Compare this to phosphorylase, which does sort of the same action as applying phosphate to something, although it does it via breaking a bond, but it does not use ATP. We're going to see phosphorylase a little later on. So our nucleotides, ribonucleic acids, adenine, cytosine, guanine, uracil, you're familiar with these. Our deoxyribonucleic acids, adenine, cytosine, guanine, thiamine, are made up of a sugar, a ribose sugar, a nitrogenous base, and a phosphate group. If it lacks the phosphate, it would be called a nucleoside, provided it has the sugar. Nucleoside, sugar, nucleotide, phosphate. For further reference, adenine and guanine are your purines, cytosine, thymine, and uracil are your pyrimidines. Now we'll move on to the carbohydrates, or the sugars. The most common one you're probably familiar with is going to be glucose. Sucrose is just a dimer of that. And what do? Well, production and storage of, we say energy, they're really storing electrons that can be accessed to generate energy, because these are not energetic molecules in the same way that ATP is. They also are good for structural, such as cell walls and the extracellular matrix itself. They are used in DNA and RNA, as we just saw, the ribose sugars, or the deoxyribose sugars. They can be linked to proteins, which can signal where the protein is supposed to be. So for localization or other information, or even activity. And they can be linked together as large branch chains, especially glucose, that is going to form this molecule, which is glycogen. So the G here does not stand for glucose. This stands for glycogenin, which is a protein off of which all these little glucose molecules will extend, and sometimes they branch and they form this huge web of material. As you can imagine, with all these hydroxyls and all this oxygen, this molecule needs to be highly solvated. It needs to be kept in a great deal of water. Glycogen requires a tremendous amount of water to remain solvated. This brings us to the fatty acids. Now, fatty acids are also used in, again, the production and storage of energy. Really what we're talking about are electrons, and these can have upwards of 6 or more energy, we'll get to that later, than sugars by weight. Now, what this really means is that the electron composition of a fatty acid can generate a lot more energy via ATP, beta oxidation, electron transport, we'll get into all those later on, than glucose does. Glucose is pretty good, it's okay, but fatty acids are even better. Fatty acids play a role in the generation of cell membranes, and of course we're all familiar with the plasma membrane, the phospholipid bilayer, and all of that, and they're good for cell signaling. Especially cholesterol will derive into steroid hormones, as well as additional signal transduction. So these play a role in, of course, energy. Everything does. Some structural structures, such as membranes, and some signaling. So just to give some examples, here's our phospholipid. So we have a glycerol backbone. Usually there's a phosphate group and a couple of alkyl chains. And then here's sort of a schematic of a phospholipid bilayer. Now that we know the actual biomolecules, we can look at the cellular structure itself. And this is important because for a cell to function, it must be isolated from its environment. The environment around a cell is usually hostile to the functions that are happening in the cell. So what we have in the cell is an isolated chemical factory. That's it. For some reason, a cell established a plasma membrane and then started creating its own set of chemical reactions, which then proceeded to replicate and stay in existence. It's an evolutionary event. So eukaryotes, being quite a bit larger, also create additional compartments. Now, this is not to say that prokaryotes don't make compartments in air quotes. Prokaryotes do, they just use the plasma membrane as kind of their metastructure for doing that, whereas eukaryotes will create discrete independent compartments to house pretty nasty chemical reactions that the cell does require to function. So for example, here's sort of a generic eukaryote cell, and we see the Golgi, which is just an extension of the ER, which really that should be associated with the ER. We have the rough endoplasmic reticulum, where ribosomes will be found for proteins that will be secreted or stored in membranes. There's also the smooth endoplasmic reticulum, not shown, in which we will have fatty acid synthesis and detoxification. The mitochondrion, which that's a singular form of the word, is where we house the primary ATP synthetic apparatus, as well as heme biosynthesis and urea biosynthesis. A lot of things happen in mitochondria. The lysosome is the site of cellular digestion, and this, along with the mitochondria, has some slightly different pHs. The pH of the lysosome is roughly 5, as we'll see, and the mitochondria has a couple of different pHs, a little bit more basic in the matrix and a little bit more acidic in the intermembrane space. So lysosomes I want to focus on for just a moment. These are single membrane organelles. They have an interior pH of 5, and they are the site of cellular digestion, including other organelles, such as mitochondria. So lysosomes will digest mitochondria. Now this is a controlled process called autophagy that the cell does as mitochondria age. So to give you an example, here's a mitochondrial cell that can be absolutely enormous. Here is the nucleus, just to give you an idea of how big a eukaryotic cell can become. Cytoskeleton, I'm not going to focus heavily on this. So what is it? Well, it's the microtubules, actin, intermediate filaments that give you motility using the plasma membrane, the shape, and cellular organization. A lot of our organelles are associated with the cytoskeleton, and so the cytoskeleton helps keep everything together and in position. The cytosol, we see metabolism of carbohydrates, amino acids, nucleotides, synthesis of fatty acids to some degree, although that's also going to occur in the smooth ER, and it is our primary site of protein synthesis. I don't want to overdo this, but just to give you a grounding, you can see that actually the volume of the cell as depicted here really is mostly stuff and not mostly water. It's mostly membranes, it's mostly ER, but we also have mitochondria that are all bound up in the cytoskeleton, have our plasma membrane, our rough ER, some microtubules along which we'll have our kinesin motors moving along, microfilaments which will help give some structure to the plasma membrane, our intermediate filaments that help with flexibility. And now on to something pretty important. This is our first disease for this class. We will see diseases in almost every lecture because almost every system can fail in some way as to give a disease result. Now, what I want you to really understand is that these diseases are dangerous and they're also mostly childhood diseases because remember, these are metabolic systems, and metabolic systems have to be functioning from the moment of birth, if not earlier, through adulthood. So these diseases definitely appear in children, and so we need to be very, very careful and very, very thoughtful when we're diagnosing and examining children. So we're going to look at lysosome failure. So in the lysosome, which is our primary site of digestion for the cell, there are all kinds of digestive enzymes, just huge numbers of them. I'm going to focus on two of them. Lysosomal acid lipase, the key here is acid lipase. Lysosomal is just telling you where this enzyme is found, and cholesterol ester hydrolase. So both of these involve the removal of fatty acids from either triacylglycerol or from cholesterol esters. So pretty much we're just hydrolyzing cholesterol esters to cholesterol and fatty acids, or we're removing fatty acids from triacylglycerol and releasing glycerol. Okay, so this is vital. This is important in the supply of cholesterol to cells for growth and membrane function. This is a big deal. Okay, we can make cholesterol. It is a very difficult process for cells to do, as you will see. We can make fatty acids, but it is a highly energetic and material demanding process, so mostly this comes from diet. And since these things are ubiquitous in diet anyway, we have to have enzymes that clear these materials when they're introduced to the system. Consider for a moment, although it's only tangentially related, insulin. So you intake sugar dietarily, and insulin removes the sugar, pulls it out of your bloodstream, puts it into cells where it can be stored as glycogen or degraded for energy. Now, the big deal here is we've introduced a material, we have to clear it out. We need to get the system back to its standard or steady state. That also happens with fatty acids and cholesterol. In fact, everything we do that changes the system responds to undo that change. That's steady state. So what happens? Well, you'll see these symptoms. Massive accumulation, usually exogenous cholesterol esters. Exogenous just means from outside of the cell. And you're going to see cholesterol esters basically everywhere, especially the liver. It's going to potentially lead to extreme liver damage, but you're going to have cholesterol esters in pretty much every organ system. So we're talking about cholesterol ester hydrolase. But a similar problem can arise from lysosomal acid lipase, but it's just with triacylglyceride. This gives us two major diseases. Wolman disease. This is lethal by age 1. That's 1 year. And cholesterol ester storage disease, which will result in hepatic failure and atherosclerotic vascular disease. Now, here's the thing. These two diseases are essentially identical. One is lethal within the first year of life, and one can be treated. So let's look at this. What is the difference? Well, the difference, amazingly enough, has to do with enzyme levels. In Wolman's disease, there is no functional enzyme detectable. None. Zero. There is no enzymatic activity of the cholesterol ester hydrolase, or even the lysosomal acid lipase. And this comes because there's a premature stop codon in the mRNA transcript. So what happens is the enzyme isn't even synthesized. You get a failed amino acid sequence, which is then simply degraded. So there is no enzyme. You can't find it. You will see no activity. However, cholesterol ester storage disease also has a genetic mutation, a single G to A, which results in a failure of the enzyme to remove an intron. Now, introns are kind of RNA sequences that are removed so that only the exons are exported from the cell and then translated into protein. So what this results in is a protein that is larger than it should be. It has more amino acids than it should have, which disrupt its folding, which ultimately disrupts its activity. But you have some activity. Somewhere between, I believe, 5 to 12% activity is detectable. Some activity. So notice the difference between the two is roughly 5% enzymatic activity, and that both give us these symptoms. Wolman's is lethal by age 1. Cholesterol ester storage disease can be managed, and it's a difference of just a few percent of enzyme activity, from no detectable activity to low detectable activity. So how do we treat it? Well, therapy for Wolman's disease is basically untreatable, although there are some experimental treatments that are being explored and researched. But CESD, if caught, is perfectly treatable with dietary control and some pharmacologics. So these individuals can lead a normal life. Again, with careful dietary control and pharmacologics. Wolman's disease is untreatable, and that brings us to a problem. If you have a very, very young patient, let's say 6 months old, and you see these symptoms, what do you diagnose the patient with? And I'm going to give you a moment to think about it. If you said Wolman's disease, you are wrong. There is absolutely no way to diagnose this as Wolman's disease with just these symptoms. If you said cholesterol ester storage disease, you are wrong. There is no way to diagnose this disease with just these symptoms. In other words, you can't tell which disease it is until you have done the enzymatic activity assay, which will probably require some sort of liver biopsy would be best in order to examine the actual lipases and cholesterol ester hydrolases in the liver. You can also do this through genetic screening, so you could do a genetic screen, but just the appearance of symptoms does not inform which specific disease. It merely informs which class of disease. In this case, some sort of lysosomal failure. Now, there are a large number of lysosomal failures. One of the more commonly known ones is Tay-Sachs, which has to do with ganglioside metabolism. They all have their own names for what is not being properly digested. And that's really it. So now you should have a pretty good overview of the cell and how it works, as well as what are the major players in terms of biomolecules. And we've also examined our first disease, and you're beginning to see how tricky diseases can be to distinguish from one another, because you're looking for a specific and often small detail amongst a group of diseases that have very similar symptoms.